Hello everyone. Thank you so much for stopping by to watch this video. I'm very excited today to walk you through the first study of its kind, where we basically characterize the localization of soybean vein necrosis virus proteins in the plant cells. So first of all, I would like you to know that soybean vein necrosis virus, or SVNV, was first reported in Tennessee in 2008 by Dr. Xanthakis. And this virus have a single-stranded RNA genome. It belongs to the family called Boniaviridae, order Boniaviridae in the family Tassoboviridae. So basically, these viruses are so cool because they basically can replicate and behave in two different systems, completely two different systems, the insect or the animal system and the plant system. So they can cross kingdom. So they're super cool and I love working on them. However, their genome is very complicated. So when we look at their genome is composed of three segments, S, M, and L. On S segment, we have the NSS or the silencing protein. And as it's called, basically it helps the virus to silence the plant system so it can infect it successfully. Also, it has the capsid protein, which basically encapsulates the virus RNA genome to help it to be protected from the cell. And then on the M segment, we have the movement protein, and as it's called, basically it helps the virus to move from cell to another cell through tubule formation, which is so fascinating. Again, we also on M segment, we have the GN, GC, or the glycoproteins, and these are very important for the insect acquisition. Basically, they are found on the outer shape of the virion. Finally, on the L segment, we have the RDRP polymerase, which basically very important for the virus to be able to replicate from negative sense to positive sense so the plant can then replicate it. So why we study this virus? First of all, SVNV is a threat to soybean in the United States and also in other parts of the world. And soybean is very important for us for sure. And understanding the virus localization or how it's behaving in a cell is very important for us to, in the future, to develop maybe more important methods, management methods on a local level. So this is very important for us. When we look on the phylogenetic tree here, which was basically determined or constructed based on the N protein, we see that SVNV have a very interesting clustering. It's not grouping with other with the big groups for orthotasboviruses. It's only clustering with necrotic mosaic virus. So this is very interesting. And the virus causes symptoms of necrosis, which basically it's a cell death that appears between the veins of the infected leaves. So these are the symptoms that are associated with this virus. Our hypothesis is that SVNV will exhibit similar but not identical localization patterns to its characterized family members. Basically, based on this phylogeny and how we see SVNV is clustering alone, we expect that it would have some identical or some similar localization patterns to the characterized one. So the characterized ones basically are TSWV, INSV, CACV, and IYCV. These are the ones that we know a lot about their localization, and we've done it before. However, SVNV has never been localized. That's why I said it's the first study of its kind. So the objective is first to detect, actually, it's just one big objective, is to detect the localization pattern of SVNV proteins and compare it to other characterized orthotasboviruses. The methodology that we use is what we amplify all the proteins except L with the PCR, and then we clone them in penter. We send them for sequencing, and then to confirm that the sequence is good. And then we used some tools to analyze the domains of NSM using the Muti Finder and the AlphaFold. And then we've used them with the GFP in both C and N terminal, as we see here. And then we moved those constructs in Agrobacterium tumefactions for infiltration. And then we infiltrated benth or Nicotiana benthamiana. It's a plant model, can be infected by SVNV. And then we finally used the confocal to take images. So when we look here, the first column is GFP fused to the protein of interest. Second column is RPH2B, which is a plant marker that have the nucleus fluorescent as red. And then here we have an overlay. So the N protein localized into the cell periphery and inside the cell nucleus. The NSS protein also localized in the cell periphery and inside or weakly inside the cell nucleus. GN and GC, both of them localized to the cell membrane and nucleus membrane, which also agree with their domains because they contain transmembrane domains, which triggers them to localize into the membrane, basically embed the protein into the membranes. So we were expecting that. We were not expecting it for N for sure and for NSS. And then finally, the GFP, which is the control, localizing into the cell periphery and the nucleus. Okay, so if you look at this figure, we are missing one protein, which is the NSM. So what's happening? So basically, I tried localizing NSM protein multiple times, at least 6 or 10 different experiments, replicates, and every single time I have never seen expression for the protein. Of course, we sequence the plasmids, we make sure that the protein is there, it doesn't have a stop codon, it's normal, no mutations, it's fused to GFP, so why it's not expressing? And then at that time, we had sequences from Alabama, from the field, and we found that NSM have mutations in the field. So we wanted to localize them together and see if we're going to see any variations or not. So NSM of Tennessee, and we call it the wild type, and also all these proteins that are localized here also from Tennessee strain. This is the first strain that was first deposited in 2008. So the wild type never expressed, as you can see here, at 2 days or at even 4 days. But when we look at the NSM, the one with a mutation, and I will show you the mutation afterwards, it was able to express. Our control was another protein from the virus called the nucleocapsid. This is the one there on top here, and we know that it can express. So when I told you that I expressed the wild type NSM, but it never expressed, I started observing symptoms of cell death on the infiltrated plant leaves. And that led us to use something called cytotoxic blue stain to determine if we have cell toxicity or cell lethality with NSM or not. So basically, cytotoxic blue is a very easy way to determine cell death. You infiltrate this stain 30 minutes before imaging, and if you can see it inside the cell wall, it indicates that these cells are dying or they have some kind of toxic proteins in them. But if you can see the stain outside the cell wall, it means the cells are still alive. So when we look here at 2 days post-infiltration, we see that the wild type does not cause cell death because you can see the stain is all outside the cell walls. And also in NSM, the mutation, the mutated one, does not also have cell death. But when we look at 4 days post-infiltration, this have already changed because the Tennessee NSM is now have cytox inside the cells, indicating cell death. However, the NSM Alabama, one with one mutation, does not cause cell death even until 4 days post-infiltration. So when we look at the maps or the domains of these proteins, so first of all, they both contain a domain called DUF4830. Basically, it stands for domain of unknown function. We don't know the function of this domain. And it's more interestingly, it's coming from bacteria. So this is very interesting, and we don't even know what is the function of this domain. But we do know the function of the domain 3A. This is the tubule formation domain. So this is the wild type that I showed you, and this is the mutated one. This is from the field. We did not make this mutation. So we have a mutation here where the valine changed to isoleucine at position 143. So this mutation have caused changes on the prediction. So when we use the AlphaFold to predict the folding of this protein, we found that this mutation have caused conformational change on the protein. I know it's a very weak confidence here. However, seeing differences between the two proteins indicate that there are indeed some differences between the protein folding in both of them. So the Tennessee one is not folded properly to itself. And this basically leads to usually degradation and losing function. If the protein is not folded very properly to itself, it basically being targeted by the ubiquitins and some other degradation enzymes or proteins from the host. So maybe this is why we were seeing cell death with NSM Tennessee. But the NSM of Alabama was one mutation, was showing different folding, and maybe this is why this protein was able to express, because it has a proper folding, it can do its function, and it doesn't result in cell death early. Maybe this is why. We don't know if this is the true reason, but this is what we saw. So when we look into this as a bigger picture, we basically found that SVNV proteins have a stronger association with the nucleus, and this is very unique. Only the capsum chlorosis virus, which is another orthotasbovirus within this clade here, was able to localize in the nucleus, but this was only NSS. SVNV showed that it's N, NSS, and NSM localize all of them in the cell nucleus. And this indicates that SVNV is unique within the orthotasboviruses and might maybe have a different infection cycle or replication cycle. Maybe it have a stronger association with the nucleus. And we hope in the future we investigate more of that using the protein-to-protein interaction. And we also shown that SVNV causes cell death early in infection in the wild type. However, in the mutated one, it causes cell death late in the infection, allowing for the virus for more successful infection and also for more successful spread. Because if you kill the plant cell early infection, you're basically limiting your chance to be spread by the NSM and also to be picked up by the insect to be spread to other plants. So this was very cool for us, and we were interested in when we saw these results. And of course, I advise you to read the full paper. It has way more than what I just presented. It has localization in insect cells. It has a lot of sequences, phylogenetic trees, and it have co-localization, and it's cool. I would invite you to look at the paper and leave some feedback. Thank you so much. Of course, I would like to thank all the amazing funding agencies, Auburn University, and the soybean producers. Thank you so much for listening. And please, if you have any question, ask my PI for my email, or maybe I can just put it on the poster now before I print it. Thank you so much.